Welcome to Mike 314. This is the first video in our series of review videos this semester. My name is Sean Dunn, and with me I have Jesse Tier, your TA. This review is intended as a supplement to the lecture. While the review will be fairly comprehensive, it is not a replacement for attending lecture or taking good notes. Okay, that being said, so to go on to our first point, let's take a brief look at the learning objectives. This is our learning objectives for the cell bio, which Dr. Jacobs presented Wednesday, January the 10th. So we want to look at how information is stored in the body, in the cells of the body, and it's done by DNA. And we'll talk a little bit more about what DNA is. We have to be able to take the DNA. We have to take the information from the DNA and access the information. Well, I'm not going to read all of these, but basically, this is the learning objectives we're going to cover today. Okay. Okay, so I know for some of you, you're not biologists. Last time you took it was in high school and you hated it. So we'll just start off briefly by saying that the cell is a small piece, or your body is made up of many cells. Inherently, it's the smallest form of life in your body, and you're made up of something like 10 trillion cells in your body. Very small is basically a takeaway point. And I really like Dr. Jacobs' analogy where if you were the size of Phoenix, the cell would be the size of your body. Okay, so let's go ahead and start by looking at what the master blueprint is. The master blueprint is inside of your cell, and it's basically the DNA. It will tell your body what it needs to do depending on the situation or environment that it's in. So if it's a really hot day and your body's overheating, your DNA may open up this blueprint and tell your body how to react. You know, your body needs to sweat. It needs to maybe shed hair if you're very hairy. So, okay. What more? Okay. So this blueprint, we need to keep it in a safe. This safe that we keep the blueprint in is called the nucleus. Now, the reason why the safe is important is because if anything were to happen to the blueprint, the cell would basically die. So it's kept in the nucleus, and then everything outside of the nucleus is called the cytoplasm. And the cytoplasm is basically the workhorse. It's basically the working environment. Now, the safe is made up. The safe protects the DNA of the nucleus, and it's made up of an oily membrane. And it's hard for things to get in and out of the membrane, but it does happen. So, okay, so we have the nucleus, we have the cytoplasm. And is this, okay, yes, and inside the cytoplasm is where everything happens. We have our factories where proteins are made. We have the trolleys, the buses. We have everything that needs to happen with the cell in the cytoplasm. And the nucleus is just the safe. Okay, so moving on. So again, you're the size of Phoenix. Your cell would be the size of the bedroom. So the nucleus, the safe where the blueprint is kept, would be the size of your closet, and the DNA is the 60-mile long string, the width of your hair. Okay. Again, we have our oily membrane here, and yeah, we pretty much covered most of this. The DNA is made up of the letters, the A, T, G, and C, and we didn't really go too much into the biochemistry of DNA, but if you have questions, we are a very good resource to ask. So you can bring those to us. Okay, yep, and to the point of cell machinery, again, it happens in the cytoplasm. The nucleus is where the DNA is kept, but here the cytoplasm is where everything else happens. This is where the proteins are made, such as the shoes for the T helper cells or the macrophages to run to the site of infection. Many other things. Again, we're pretty brief on the biochemistry, but if you're interested, we can definitely go deeper into that. Oh, and the proteins, just again for a perspective, protein would be the size of a dot in comparison to U, the size of Phoenix. So we can pack a lot of proteins into a single cell. If we need to send out proteins, we can definitely do quite a bit of those. Okay. Okay, so satellite dishes. Okay, so as you what happened here? Okay, so we mentioned before that we have a nucleus with DNA in it, and this nucleus is surrounded by a membrane, an oily membrane that protects the DNA from the cell, from the cytoplasm, where all the cell machinery is. Okay, so likewise, the cell is also surrounded by an oily membrane. Now, in order for the cell to function, it needs to be able to communicate with the outside environment of the cell. So things need to get through this oily membrane, which is not an easy task. In order for this to happen, the cell needs to have satellite dishes, cell receptors, I think is the term. So these satellite dishes, they receive signals from the outside environment and tell the cell what to do. One really important satellite that you're going to repeatedly hear throughout the semester is going to be the CD4 satellite. Now there are many other satellites such as CD8 like on the T killer cells, or CD20 on the B lymphocytes. So just keep that in mind. This will be a repeating theme. We're going to talk a lot about these satellites, and especially the CD4 satellite. Okay. So this is the lecture from, what was this Wednesday? This is still Wednesday. Okay, so we're going to start getting into the immune system here. We're still in non-specific immunity. So we start out with the nose cell, and a flu virus. You're basically going to inhale a flu virus into your nose, and this flu virus is going to insert itself into the cell. It's going to get past the cell membrane, the oily layer. So the cell is infected. What does the cell do? Well, the cell can't do anything without accessing the blueprint. The blueprint is the most important thing. So it's going to look in the index, and it's going to look at pages, say 10 and 38, and say these pages will tell the cell, here's what you do when you're infected with flu virus. Now each page does something for a different circumstance, but pages 10 and 38 in this instance is how you should react to the flu virus. So the cell is going to make many copies of pages 10 and 38, about 1,000 Xerox copies. For you biologists out there, of course, we know this as messenger RNA, but for our needs in this class, we're just going to call them Xerox copies. And again, some of our terminology in this class is a little bit funny, but it's on a tangent here. We'll digress a little bit. It is really important to be able to explain complicated biological ideas to non-science people. So hopefully you can take away some easy ways to explain this, explain the biology of HIV, to explain to the general public.